Okay, so now later I'm going to show you a basic process first. Okay, I'm going to show you the basic process, what is meant by complementary base pairing between the two ends, okay, between the target DNA and the vector. And also what is within the selections of the registration enzymes, and what is the issue orientations here that will make us, okay, prefer to use the sticky end compared to the blunt end. But both, they have the pro and cons. I will summarize it later. Okay, so now I'm going to share screens from another device to show you guys the process now. Okay. Wait a while, let me share. Okay, can you see this? Okay, can you see my share screen now? Okay, so now I'm going to go through this process slowly. So just look at this, how this animation actually work. Now, if you look at this, the one that I highlight with the yellow color, okay, can you see that? The one that I highlight the yellow color. So this yellow color actually is a restriction site. Can you see that palindromics? GGA TCC. GGA TCC. So these are forward directions and reverse direction. GGA TCC. So GGA TCC, GGA TCC. Can you see that? Okay. So now, the one I highlight with the blue color, the light blue, is a gene of interest. Okay, I simply make up. Okay, we don't, don't go and find what is this gene, doesn't matter. So what we're going to have here, GGA TCC, the registration enzyme that recognize this registration site going to cleave between G and G. You're going to cleave between G and G, G and G. So produce, okay, G and G, G and G. So break the phosphodiester bond between G and G, G and G, G and G, G and G. So what we're going to do here, we're going to get 3 fragments, right? The first fragment here, second fragments, and third fragment. So I make it, can you see that? 3 fragments. Can you see that? We have 3 fragments here because we use the registration enzyme. So this is the gene of interest. Okay, so if I want to make this to glue on this, let's say for example, another DNA, okay, another DNA, we call vector here. Okay, so this vector, if I go and search it, oh, I saw the same. It must be the same registration site. Can you see that? GGA TTC, GGA TCC. Cannot GGA TCC. Can you see that they are the same registration site? So if they are same registration sites, I'm going to do here is I'm going to cut it with the same registration enzyme. I have to use the same registration enzyme. I cannot use a different one. If a different one, I won't be able to get the complementary one. Correct enough? Okay, so if I cut it here, can you imagine? I cut, okay, also recognize between G and G, cut it here, remove this phosphodiester bond. Again, now I can actually break this DNA into 2 because only one registration site, I'm going to get 2 fragments. So what I'm going to do here is now, if I mix them together, do you realize that the gene of interest that I have now, the sticky end, they are complementary. Can you see that? Complementary. Can you see that? Complementary. So when they are complementary like this, then they can form the hydrogen bonds. Means that we can actually put in our glue here. They can hold, right? Hydrogen bond can help us to hold. So effectiveness definitely higher. Are you clear? Effectiveness definitely higher. But what is the problem, or what are the problem of the sticky end registration enzyme? We have 2 main problems here if you use the registration enzyme with which produce sticky end. So what is the main problem? The main problem here is because they are sticky end, when I cut it here between G and G, G and G, before I add my gene of interest, they can glue back. Can you see it? It can form back. So this process is called religation. RE, because we cut it already, before I paste a new one in, they fall back. Can you see that? They close back. So this one we call religation. Very, very often actually take place when we use the registration enzyme, okay, very often that we use the restriction enzyme where they produce sticky end. Can you see that? Okay, but right, I want them to be open up so that I can put in my gene of interest here. Then I can apply the glue. Okay, I can apply the glue, I can apply the glue, or DNA ligase can help us to seal back or form back this phosphodiester bond. Can you see that? But the thing here is, because we use a sticky end restriction enzyme, so it means that they can re-ligate back. So when they re-ligate back, sorry, we can't paste our gene of interest anymore. Can you see that? Okay, that is one of the disadvantage. Okay, second disadvantage, because we want to make sure that, can you see that? We want to make sure that this end and this end, they are complementary. So it means that we have to make sure that they match. Can you see that? This one, the restriction sites at these three locations must match. If they don't match each other, then we can't. Are you clear? If you use a different one, then you won't be able to glue it together already. Try to imagine here, if let's say, okay, here, let's say, it's not GATTC anymore. Let's say GGTC, for example. If GGTC, I cannot match it because G and T cannot form the hydrogen bond. Can I see that? So it means that we have to make sure that the restriction sites must match first before we can use. Are you clear? Before we can use. So these are the two, what we call the limitation. First, when you cut it, it produces sticky end, it's going to ligate back. So this process is called religation. This is the first problem. Second problem is, we have to make sure that we must search the entire length of the DNA and make sure that the restriction site must match. So when the restriction site match already, then we use the same restriction enzyme to cut, then the sticky end produced here will be complementary to each other, allow us to join. So these are the two limitations. But what is the benefit? What is the good thing? The efficiency is very high. Why the efficiency is very high? Because sticky end, the hydrogen bonds can help this complementary to hold this complementary sticky end together, allowing the ligase later. Ligase is a glue, okay, to seal the sugar phosphate backbone by forming the phosphodiester bond. Okay. So now, how about blunt end? What is the problem if we have blunt end? If we have blunt end, then we can blunt end definitely, because as I said just now, okay, blunt end. Basically, if you use a blunt end, what will happen here is you do not need to actually have same restriction site, because we can use two different blunt end restriction enzyme to cut it, doesn't matter. Okay, you can use two restriction sites, blunt end restriction site, cut it, because at the end, blunt end, they do not need to have the complementary base pairing. Can you see that? Are you clear? But the blunt end problem here is what? Blunt end problem is the effectiveness will be lower. You try to imagine, as I just now say that you want to glue two rods together, but no one hold it. So it's difficult for us to glue it. Okay? So this is the first one just now I say that. Why we need to use the same restriction enzyme, why we need to make sure that the restriction site must match for sticky end, but not for blunt end. Blunt ends freestyle. As long as two blunt ends, no problems already. So what is the limitation of blunt end? Blunt end, basically, the effectiveness will be lower. Okay? So just now we talk about orientations. Now, what's my orientation? Orientation is very, very important. Think about the protein synthesis when we want a transcription process. Transcription, they always check one strand only. If let's say I use the top strand to read it, if I read the top strand to produce my mRNA and polypeptides, I read it ATT, CCC, GGG, TAA, CCT, and so on. But because of complementary like this, if reverse, can I see? If I reverse it, if I glue it this way, do you realize that I still can glue it together? GNC, TNA, TNA, GNC. But now the sequence becomes GAG, GTT, ACC. So it's going to produce a different polypeptide for us. if this we call the reverse orientation. In fact, what I want to glue it this way. Can I see that? I want to glue it this way. If I glue it this way, then correct, ATT, CCC, GGG, TAA, and so on. But reverse, because this one will happen 50-50%. It's a random process. We can't control. Can I see that? We can't control. Can I see that? We can't control. So in this case, if I read it now, it becomes GAG, GTT, and so on. So you're going to produce a different polypeptide for us. Correct? Okay. So in order to overcome this, sticky end can help us to overcome, but the process will be more complicated. Are you clear? I will show you, but do not need to remember. Exam question come out before. But just need to know a brief one only. Question won't ask you to do this. Okay. So what you need to know here is if we want to overcome the reverse ligation like this or opposites ligation, then we have to use a sticky end. Blunt end cannot do anything. I will show you the process now. Now look at this. This is my gene of interest. Oh, I forgot to highlight. Okay. Another one is AT. Okay, so can I say I highlight it with two different colors? Why highlight two different colors? Basically now I'm going to cleave my DNA with two registration enzymes. One registration enzyme recognizes G and G, another one is A and A. So I'm going to create this sticky end. But can you see that these two sticky ends different? Can I see that? So another molecule also, I need to make sure that yellow with yellow, green with green. So I have two different registration sites. So I cut the registration site number 1 with enzyme 1. Cut the registration site number 2 with enzyme 2. Then what I'm going to produce? In this, I'm going to produce 3 fragments also. But this fragment is actually free, so can remove. Okay, this fragment free can remove. So this process can help us to overcome what? Now, if you want to religate back, they can't already. They can't religate back. Why they can't? A and C are not complementary, G and T are not, C and A, T and G, they're not complementary. So no religation now. Okay, and I want to put in my gene of interest, it must be in the correct orientation now. Can I see that? G and C, A and T, T and A, G and C, A, T, G, 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 C, C, G, T, A. If I reverse, can they bind? They can't bind anymore. So we can solve the problem of the reverse orientation. We can solve the problem of religation, okay, by having two different registration sites. But we have to make sure that the same registration site, we have to use back the same registration enzyme. Then I can overcome the reverse ligation. Can you see that? So no matter how, I have to make sure that it can help us to make sure that the ligation is in the correct orientation. So this is just extra information for you guys to know we can overcome. But blunt end, no matter how, we can't do anything because they are blunt end. No matter how you turn, it's still blunt end because there is no complementary base pairing involved. Okay, so I stopped the share screen here. I'm gonna share it here. Okay, so do you guys have any questions? Yep. Okay, so basically I'm going to summarize here. Okay, so if you put it here, pro and cons for sticky end. Definitely, okay, the effectiveness is high. Efficiency, okay, effectiveness is high. Okay, for blunt end, will be low. Oh, sorry, it's a pro. Okay, so effectiveness is high, but what is the problem? What is the cons here? Basically, religation takes place, and we need to use the same registration enzyme. So blunt ends for pro, no religations. I mean, religations will be low because blunt end low. Okay, and we do not need to use the same registration enzyme, not necessary. But what is the cons here? The effectiveness lower. Okay, so another pro here for sticky end, we can overcome reverse ligation. But here, cons cannot overcome. If you use a blunt ends, you cannot overcome reverse ligation. Okay, basically this is the summary for the sticky ends and also the blunt end. Okay, so this is what I mean here. So for the insertions of the target DNA fragment in the plasmid vector, it's preferred to use two different registration sites to digest the DNA so that the different sticky end are generated. So the two different sticky end can prevent the religations and allow the foreign DNA fragment to be inserted in the directional manner. So what is the advantage of the blunt ends? Basically, they does not require any registration site in its sequence. Are you clear? Means that I can use a different one and not a specific one. So blunt ends usually generate in the polymerase chain reaction. This one we're going to learn in the next subtopics. Okay, and PCR generated blunt end foreign DNA fragments may be ligated into the blunt end vector generated from the registration digest. Okay, so at this moment you'll be confused. It's okay. So we're going to talk about this in PCR techniques. Okay, in the next subtopics. Okay, so we continue with this molecular vector. Then we're going to have a rest, right? So what's mean molecular vector? As I said, that once you cut the DNA out already, when you cut the DNA out already, then what we're going to do, we want to paste it, right? But the thing here is how we transport it into the host cell. We can't, okay? So basically, we need to have this molecular vector. So this molecular vector is a DNA molecule used as a vehicle to artificially carry foreign DNA material into another cell where it can be replicated or expressed. So replicated here means clone. Expressions means that to form protein for us. Okay, so vector, we can take it from the virus, bacteria, or even the cells of the higher organism. So the things that you need to know, this highlights, what are the features of a vector? All vector must have these features. First, they must have the origins of replication. This is very important. They must have the cloning sites, or what we call the multiple cloning sites, and selectable genetic markers, as well as they must have promoter. But promoter depends on what kinds, okay? Depends on what kinds of the vector we are going to use, okay? So we have two kinds of vector. We have the cloning vector. Cloning vector idea here is to clone the gene, but not express, Means that I don't want protein. So if I don't want proteins, then yes, you do not need to have. So they do not have promoter for the inserted gene because I want to clone only. So what's mean clone? Means that, okay, if I have one copy of the gene, I want to clone them into multiple copy. I can ask bacteria to do this for us. It's an old technique. So cloning vector is an old technique. We use a bacteria to clone a particular gene for us so that I can get multiple copies. Let's say I have only one copy. I want to get 10 copy, millions copy. How? We ask bacteria to clone for us. So in this case, we don't, our intention is not to produce protein, right? So we use a cloning vector. Are you clear? We use a cloning vector. So we only store genetic sequence, clone the genetic sequence, okay, and also store it. So do not need to have the promoter. So therefore, they are incapable of allowing transcription and translations of the inserted gene. So they won't produce protein products. Okay, if you want to have transcription and translation, then you must use this, what we call the expression vector. I forgot to underline, so please underline for me. Okay, expression vector. Expression vector, then it can help us to transcribe and translate the gene because they do have the promoter. So what is the idea? What is the function of promoter? Promoter is the site for RNA polymerase to bind. If you have gone through my lectures on these gene expressions, okay, or gene control, RNA polymerase will bind to promoter. So when they bind to a promoter, going to initiate RNA or mRNA synthesis. or we call it as a transcription. Are you clear? Okay, so we need to have the promoter. So promoter is very important, okay, because it's a site for RNA polymerase to bind and then initiates the mRNA synthesis or transcription. So it means that if the vector or this molecular vector, they do not have the promoter, then sorry, it becomes cloning vector. They can't produce the gene products for us, okay? Promoter is the must. So this a lot of questions asked about this. What is the characteristic of or features of the vector? So these are the answer here. So first, we need to have the presence of the original replications. It's very important. Why? Because if you put in this vector into the bacterial, we need bacterial to propagate for us. We need the bacterial to produce more for us. So original replication is a sequence of DNA at which replication is initiated on a vector. So without this, we won't be able to clone. Okay, you try to imagine, sir. This is one bacteria. Yeah, they take in the plasmid. So this plasmid or this vector contain the gene of interest that we have. But the problem only one. How we make it multiple copy? If you want to make it get multiple copy, then this vector must have origin of origin of replication. So here is origin of replication. So with this origin of replications, then you can see that now bacteria can produce many copies, and each copy we're going to have our gene of interest and also origins of replication. And then this will act as template and continue to produce even more. And then we can actually let the bacteria to carry out binary fission. Then we have more bacterial cells to produce the proteins for us. Okay. Now, what is mean cloning site or multiple cloning sites? So cloning site basically they contain unique restriction sites so that it can be cleaved at this. So it means that if this is my plasmid or vector, they must have some point at this vector allow us to break it and then put in our gene of interest later so that it can transfer it into the bacterial cells. Okay. So what is the functions? The restriction site in the multiple cloning sites are first cleaved by the restriction enzyme, and the target DNA sequence can be inserted into the vector. If we can't cut it, how we paste it? All right, we cannot paste it. Are you clear? Okay. And they must have what we call selectable genetic marker and also the reporter genes. So at this moment, what you need to know, I will go into the detail about this genetic marker reporter genes later. Okay, maybe tomorrow. So it helps us allow the selections of the positive transform cells. Okay, so if this is a bacteria, one bacteria take in the plasmid, another one didn't take in the plasmid. How we know? So genetic marker and the reporter genes, okay, on this plasmid will tell us, they will report to us. Oh yeah, success. Oh, not success. So how we're going to learn it next subtopics, not next subtopic, but basically next sessions of the topics. Are you clear? So it basically acts as a reporter, tell us, oh yeah, I success already. Okay. So at this moment, don't worry about these penicillins, ampicillin, or Lexac genes. No worry. I will come back later. Then you will understand. Okay. So presence of the promoter usually to try the transcription. So without the promoter, then definitely no transcriptions can take place. Okay. So in this case, we talk about presence of promoter. This one's suitable for the expression vector. Only in expression vector. No need for the cloning vector. Cloning vector, we want to clone only. We don't want to have the protein, don't need to have transcriptions, therefore no need to have the promoters. Okay. So the four major kinds of the vector, so plasmid and viral vector, but our syllabus will focus more on the plasmid. So in the later parts of the topic, we're going to look at the PBR322, PUC19, and PGRO. So basically, they are double-stranded DNA. So suitable for gene cloning and also the gene expressions. The rest, you can read it, okay? Nothing special. So this is for your information only. for your informations only. You can read, if you want to know more, always can come to see me, or you can search more. Okay, the difference here actually basically is a size limit. If you want to put in a long gene, then maybe plasmid cannot, you have to use a bacteriophage or cosmid. Okay, if the size is very, very small, we always can use a plasmid. Okay. So the most commonly used vector, as I said, that is a plasmid. We're going to look at plasmid PBR322. What are the features? First, they have genetic marker. Write down. So this set of genetic marker will allow us to identify later who successfully take it. So they have the origins of replications. Can you see that? Okay, and they have a unique registration site. I always say that at least for any vector, they must have genetic marker, registration site or cloning site, and also the origins of replication. Okay, now no need to remember, this is just for your information only. This is how PBR322 with many, can you see that? Many registration site, many registration site can be recognized by the registration enzyme. Okay, so PBR322. Another one is a PUC19. Okay, PUC19. So again, you can see that they do have this, what we call the genetic marker. Okay, so they have this polylinker. Polylinker actually is the cloning site or multiple cloning sites or registration site. So this registration site is very important for the registration enzyme to cleave, open up, allow us to paste, okay, the gene of interest, and also they have origin of replications. Okay, so you look at these diagrams, they do have the origin of replication. Okay, but when you do this, don't be hand itchy, go and find, oh, I want to use this registration enzyme to cut at origin replication. When you cut at origin replication, then this plasmid no longer can be replicated anymore. Okay, so you can cut at other place, doesn't matter, but don't cut at origin. Okay, so that's why you need to know which enzyme is suitable to be used. Okay, so when a foreign DNA molecule fragments incorporated in a vector, then we call it as the recombinant DNA molecule because it combined from two different species. So protein that result from the expression of recombinant DNA, we call as a recombinant proteins. Okay. So we come to the last one before we end, quick one. So in DNA ligase, what's the functions? Basically, they join two DNA strands together. Okay, double-stranded DNA together by forming this phosphodiester bond. So we always say that it's sealed. It seals what? Seals the sugar phosphate backbone between two DNA molecules. Okay, and this DNA ligase usually used in DNA repair and DNA replications. What they do, they actually form two covalent bonds between the 3' hydroxyl ends of one nucleotide with 5' phosphate in as an end of another. You look at this, guys. Now the whole process is for information only. If you want to know more, you can always read it. But what I want to show you here is you can see that here, one phosphate, one OH, one phosphate group, one OH group. So using this enzyme, they can help us to seal the sugar phosphate backbone. Initially, can you see that? It's not sealed, but now sealed already. Can you see that? Okay, so this process required the glue DNA ligase. Okay, so with this, I end for the first hour of the lessons. Okay, this one extra information. Then later we're going to go into the whole cells. Okay.